So welcome everyone to our Zoom press conference to announce the start of a clinical trial using a combination of drugs, one an anti-malarial and another an antibiotic that we hope will be very beneficial for patients with moderate to severe COVID-19. So we're going to record, we'll make this available at the end for everyone. And then we're also going to start up here in just a moment with an introduction of our featured presenters here today. So the first one is Dr. Michael Gordon of HonorHealth. We also have Sunil Sharma of both HonorHealth and TGen. We have Kiran Avancha, Dr. Avancha of HonorHealth, and also David Engelthaler for TGen. So Dr. Gordon, I'm going to turn it over to you and have you and then Sunil and then Kiran and Dave all follow in order there and just talk a little bit about your hopes for this clinical trial. And if you could describe it just a little bit.
Sure, thank you, Steve. So I'm Dr. Michael Gordon. I'm a medical oncologist at HonorHealth Research Institute and the medical director of the HonorHealth Research Institute. And while most of our activities have historically been in conducting clinical trials in cancer, we made a major pivot to ensure that we could utilize our available resources to access clinical trials for COVID-19 and improve what we hope will be the outcomes for the patients who are infected with this virus. The first of our trials that has launched is an investigator-initiated trial, a trial that we're conducting here at the HonorHealth Research Institute, combining the drug atovaquone, which is another anti-malarial drug, in combination with the antibiotic azithromycin. And the rationale for atovaquone's combination with azithromycin comes from the biologic studies that have been done on COVID-19 looking at protein-to-protein interactions, suggesting that atovaquone would have anti-infectious and antiviral activities. And the azithromycin is an antibiotic known to have some antiviral activities on respiratory epithelial cells, the lining of the lungs, and is also part of a standard regimen combined with atovaquone for the treatment of a different infection called babesiosis. And so what we've done is we've taken the standard regimen of atovaquone and azithromycin administered orally on a daily basis for a total of up to 10 days, longer if patients are benefiting. And the goal is to not only treat patients with the goal of looking at their clinical outcome, but with our colleagues and partners at the Translational Genomics Research Institute, focus on trying to understand the biology of how this combination may be impacting COVID-19.
Okay, Dr. Sharma, if you could tell us a little bit about how you came up with this idea.
Yeah, no, thank you, Steve. This is Sunil Sharma. I'm also a medical oncologist, a colleague of Mike Gordon, and actually we came up with this idea together, me and Mike. There have been actually several papers now published recently where, you know, a lot of scientists in the world, some of them are in Arizona, but a lot of them outside are actually trying to develop timely solutions for treatment of COVID-19. And so one of the approaches that has been taken has been to look at the structure of the CoV-2 virus and basically try to do sort of computer modeling to see what kinds of already approved drugs might actually bind to critical areas of the virus. And in reading and so there's a long list of these kind of modeled experiments, and as sort of research progresses, some of them will be taken into clinical trials. So part of several groups now have reported that atovaquone may bind to some critical areas of the virus itself and may prevent its replication. And we also know that a related regimen like hydroxychloroquine and azithromycin is being tried currently in clinical trials around the world. And one of the problems with that regimen, as everybody's probably heard of, is that it can cause some sort of cardiac toxicity. And atovaquone and azithromycin is relatively safer compared to that. And like Dr. Gordon mentioned, it's already approved and currently used for a different parasitic disease called babesiosis. So in order to help our patients, we decided to embark on this clinical trial. We are also partnering with Dr. Engelthaler, who is one of the prior epidemiologists for the state of Arizona and is a partner with us at TGen North. And I see him, he's on the video screen as well, and may want to comment on this, Steve, at your discretion.
Okay, sure. Well, Dave, you want to pick it up from there, and then we'll go to Dr. Avancha.
Sure, sounds great. Good morning, everybody. Glad to be here. And I'm Dr. Dave Engelthaler. I'm with TGen North, which is our operation up in Flagstaff, where at TGen we've been doing quite a bit of testing and analysis on the COVID-19 virus, as many of you know, for the past couple of months. It's a real honor to be working with Dr. Sharma and Dr. Gordon, the whole HonorHealth team on this particular trial, which is exciting and may not only help us find better ways to treat patients, but we're also able to bring a lot of the next generation tools and technology to this study by being able to not only confirm virus is present, but actually quantify it, essentially count the amount of virus particles that we see in patients and hopefully monitor the drop of that during treatment. If the treatment is working well, the virus should drop down to very low levels and then hopefully disappear. And we'll be able to study that as well as TGen to monitor the serologic response of patients and watch the increase or decrease of specific antibody types. So we're going to learn more about the immune response. And then we're also able to capture the virus from patient samples and do the genetic sequencing. And we can understand how the virus may actually be changing during the course of infection. This is something that is really interesting to know. And do we see some what we call in vivo evolution of the virus? And does it change? And does the treatment have any effect on the population of the virus itself in the patient?
Okay, Dr. Avancha, if you could pick it up from there, tell us what your hopes are for this clinical trial. What you hope to achieve, and where do you see this possibly going into the future?
Thank you, Steve. Can you guys hear me?
Yes.
Oh, great, thank you. That was an excellent summary by my colleagues, Dr. Sharma and Gordon and Dr. Engelthaler. First of all, we're extremely proud to establish this COVID research program in a record time. And of course, as a health system, we're very fortunate to have the likes of the Research Institute. My role at the Research Institute is I'm the Chief Operating Officer for the Research Institute and also the Associate Vice President for Innovation for the health system. When this idea was being brought up, we have not only looked into the pharmacological aspects of the drug combination, but also the formulation aspects. I mean, Dr. Sharma and Dr. Gordon and myself, when we were discussing about what's the availability of the drug and the formulation that it is available in, it's actually available. The atovaquone is available in a liquid formulation. and also the ease with which we were able to procure the drug also made some of the choices for us. On top of that, the toxicity profile that Dr. Sharma has laid out, and also as an alternative for the hydroxychloroquine, made it as an attractive choice for us. While we use this combination and prophylaxis in BMT patients, there was a very interesting fact that Dr. Sharma has brought to our attention from our colleagues at City of Hope, where they were not seeing a potential COVID infection in BMT patients. That also was a very interesting factor that we were thinking of exploring when we started using this combination. With those factors in mind, we started thinking about funding this study, and this has been supported by HonorHealth and HonorHealth Foundation and the Research Institute. It's currently being conducted at 3 hospitals in our network, both at Shea, our HonorHealth Shea campus, our HonorHealth Osborne campus, and our HonorHealth John C. Lincoln North Mountain campus as well. We're anticipating to enroll up to 25 potential patients that are inpatient in our network. We already have done first patient already received this drug, and the patient is already on the 6th or 7th day so far on the treatment regimen. And what I see as a future is we have floated this idea across a few other institutions across the country, and many of them have shared their thoughts around it and shown some positive responses towards the idea in general itself. I've received once we posted this idea on our on clinicaltrials.gov, as with any clinical trial, you have to post this on clinicaltrials.gov with contact information as such. We must have received a ton of emails from folks across the country trying to see if they would like, if they can participate on the study, either as a patient or as well as an institution. So our goal is to generate enough data, a pilot data in this 25 patient data, and then see if we can expand it into other institutions depending upon the safety and efficacy aspects of the study itself.
Dr. Avancha, why is it important that this could be administered in a liquid form?
It's a great question, Steve. We considered a few factors, and one of the factors that we were looking into is for an intubated patient, and how do we take these solid formulations and titrate it, and this is all my pharmacy background coming in to share. Would it be beneficial to use a liquid formulation better than a solid formulation from a distribution perspective? But I'll let my colleague, Dr. Gordon and Dr. Sharma take it from there.
So Steve, if I may,
Please.
A couple of things. So first, the rationale of the liquid was really about the fact that we wanted to be able to continue treating patients in case they ended up in the ICU, intubated and on a ventilator, where we could continue to administer the drug without having to change the formulation by putting it into a feeding tube or a nasogastric tube. And so that becomes an important factor given how sick the moderate to severe patients can be. But going back to your earlier question, I think that one of the things that we're hoping to gain out of this is, as Dr. Avancha was noting, that expanded collaboration. The relationship in particular between the HonorHealth Research Institute and TGen is about connecting the clinical and the translational science. And as you heard from Dr. Engelthaler, there is a tremendous amount of translational work that's being done. And the truth is that for us to be able to overcome this infection, it's not a matter of weathering the storm. It's a matter of creating a process to understand and anticipate when the next storm is coming and be optimally prepared. And what we've been focusing on at HonorHealth and in the HonorHealth Research Institute is about focusing on this trial being the first of several trials that we're opening across the continuum of patients from very early infection with COVID-19 to the more severely ill patients, so that we can partition our trial options across that spectrum. And we've recently applied for some grants, actually looking at different ways to monitor patients once they're diagnosed, and hopefully identify patients who are sicker and need intervention earlier. So I think this partnership is the crucial one that will hopefully get us over the hurdle, and our ability to expand trials, not just nationally, but really with a focus on our Arizona community, also becomes very, very important. And the concept of developing a COVID coalition where different medical centers and programs within the Arizona state and the Phoenix metropolitan area begin to come together, recognizing that there is a crucial distinction between our battle against COVID and our battle against each other. And that's something that we have to overcome.
Okay, so if you all are ready, I can start reading some questions that we're getting in from the journalists. And the first one up, is this a treatment or a cure?
Sunil, were you going to grab that?
Sure, I can take an initial stab at it, and then we'll let the actual authority here, Dave, talk about it. But basically, the idea here is that if the, with viral infections, once you can actually diminish the replication or the sort of the division of the virus, meaning the virus stops dividing, then it gives us some time for the own immune response to sort of mount against the virus. So when the viral infection like COVID-19 happens, a couple of things happen. There is a sort of a genetic response that varies from one patient to the other. And, we're still at TGen, I think part of our effort is to figure out, well, why are some people actually getting sicker than other people? But let's assume that we're talking about a person who's in the moderate to severe category, the ones that we're giving these drugs to. Then they have a aberrant reaction to the virus. So part of the treatment, part of the way the treatment would work is if it stops the virus from replicating, then two things will happen. A, the magnitude of the response, hopefully that the body can mount to kill the virus, aka the antibody response will be better, or it'll take some, because it takes some time for the antibody response to occur. So hopefully it'll give some time for the body to mount the response to clear the virus. And two, hopefully it'll decrease the amount of side effects the virus is causing. In this case, all the different difficulties that the patient's having, breathing and getting intubated, etc. So in short, the treatment should, if it works, should help to cure the virus. It's sort of a kind of a tangential answer. Dave, do you want to talk a little bit about that? Because you're the card-carrying virologist.
Thanks, Dr. Sharma. I think you did a good job explaining the fact is that drugs work in different ways, and the hope here is that this combination of drugs is really going to help with what we call it being viral static, prevent the virus from replicating, prevent it from continuing to infect new cells, rather than being virucidal, which would be to essentially go and kill all the viral particles. So the goal here is you prevent the virus from replicating, and you use all those other things in your armament, which includes the immune system and all the various facets of that to help clear out the virus during the course of the infection. So it's would work in concert with what your body's already trying to do.
Okay, anyone else want to add to that? So next question is, how is that first patient doing so far?
Mike, I'll let you take it, but my understanding is too early to tell.
Yeah, I think it's too early to know. The patient actually, when she came on, was severely ill and continues to be managed in the intensive care unit. I think that our challenge with an initial study like this is going to be integrating the translational work that's being done at TGen North with the clinical outcomes. So at this point, still in the hospital, still being managed in acute care setting, and we're still collecting our data on that. But it's too early to be able to reach a conclusion, even in her case.
Okay. Dr. Engelthaler, we're going to be doing a couple of different tests with these patients. The diagnostic test, what will you be able to measure with that test?
So the viral genetic test that we've been really using to diagnose patients, we can actually use it to count the amount of virus in a sample, in a patient sample. And what we really want to understand is both the natural history of this virus as a person goes through disease and recovers, how does that viral load drop? And how, can we measure and start to predict when the virus will be cleared. Doing that at the same time now of actual treating patients with this new combination therapy, we'll be able to see, will that virus load drop even faster? And does that match up with what's going on with patient symptom and patient recovery? So that's the genetic test helps us really quantify the amount of virus in patient samples.
And how often do you anticipate that kind of test will be done on each of the patients?
Yeah, actually, I think the trial is measuring the throat swab or nasopharyngeal swabs every few days, and then serology, which is the blood test for antibody development, every few weeks. Is that about correct, Mike?
Yeah, that's correct. I think we're measuring it at 3 days and then again at 7 days of therapy for the nasopharyngeal swabs in order to give enough time for the drugs to get in and show an effect. And then the serologies, I believe, on a weekly basis, looking at the evolution of the antibodies.
Okay, so Dr. Engelthaler, if you could go into a little bit more detail about the serology test or the blood test.
Sure,
what will you be looking for there?
Yeah, sure. One of the hallmarks of the human's response to any pathogen is to develop antibodies to that particular pathogen. And we develop different types of antibodies. We develop some that work really quick, and that helps with the kind of the initial state of the infection and helps to clear out the pathogen. And then there's also another type of antibody that usually starts to be produced a little bit later. And those antibodies typically last a long time and provide what we've all been talking about, which is immunity. We still don't know a lot about this particular virus and the immune response. We're learning every day. There's a lot of antibody testing happening right now. We don't truly understand what it means for protection and whether or not people are safe from being reinfected yet. But what we're able to do, though, is carefully, again, monitor the different types of antibody and when those antibody responses are occurring with the patients in this particular trial.
Okay, a question here. Monitoring the antibodies, would you at some point hope to be able to retrieve plasma that has those antibodies from any of these patients as they recover?
So you're raising the concept of what we call COVID convalescent plasma, or CCP, which is based upon the success of convalescent plasma in other viruses such as MERS or SARS viral episodes. And this has become an important part locally and nationally in this. So the answer is yes. We hope in general that any individual who has been infected with COVID-19 and is no longer infectious would be able to donate convalescent plasma by contacting the regional blood center. In our case in Arizona or in Phoenix is Vitalant Blood Center, and scheduling to undergo plasmapheresis and donate plasma to be used in the treatment of patients. I think that the biggest challenge that we're discussing now and beginning to tackle is who are the best patients to get convalescent plasma, and in what point in the course of their illness should they be getting it. And so while step one is developing a reserve of plasma, step two becomes what's the correct utilization.
Okay, so we have a question here for Dr. Avancha, I believe. Have these two drugs ever been used in combination before?
Yes, in other diseases, but not in COVID. We're the first in the country and in fact in the world to use this combinational therapy for COVID-19. We're hoping that we'll be able to see some results sooner.
Okay, another question. How does this treatment compare to the recently FDA approved remdesivir?
So remdesivir is a different form of an antiviral therapy. So mechanistically, it's different. Remdesivir's approval will obviously be based on its ability to shorten the length of time in the hospital, and associated with that, what we think is clinical benefit. The challenges that we face in treating COVID-19 are ultimately going to be looking at future combinations of drugs, how different therapies complement each other, and in what patient population should these agents be used. And the pending approval of remdesivir is such that we'll be using it in patients similar to this, who are more severely ill, likely those who are anticipated to need intubation, which is the group that is most at risk for dying in this disease.
And I also wanted to add that, that remdesivir, from my understanding, works in about 60% of patients. So there will be other patients who may need other therapies as well.
Okay, I was wondering if someone could explain the difference between patients who are suffering from moderate to severe forms of COVID-19 and maybe those who are sick but less so. How do you make that distinction between the patients?
This is a big challenge, Steve, and we know that about 80% of individuals who are infected with COVID-19 will essentially have a moderate to severe flu-like illness that will last anywhere up to about 2 weeks. During that time, have typical flu-like symptoms, and that's the interval of time where we tell patients to quarantine themselves after they've been documented as being infected. Most of those individuals go on to do well. There is a subset, the individuals that have been identified as high risk over the age of 60, diabetes, uncontrolled hypertension, those with underlying illnesses that may suppress the immune system, who could evolve beyond the scope of just that typical flu-like illness to something more severe. And then there's that 15% of patients, roughly, who at presentation have a much more, I'll use the term malignant progression, where their shortness of breath, respiratory illness gets significantly worse rapidly. And those individuals end up presenting to the emergency room with shortness of breath and findings on chest X-ray or chest CT scan of a viral pneumonia, and ended up needing a more prolonged hospitalization.
I see. Go ahead. Could it be at some point in the future that you might also apply this drug combination to patients with a milder case of this disease, ones beyond, the moderate to severe category.
Hi, this is Dr. Sharma. So yes, I think Guy and Mike are discussing maybe with the idea of amending this protocol to allow milder cases to go on. There is actually an objective scoring system that we use based on the patient's oxygenation status and temperature, etc., to come up with the exact definition of who's moderately or severely ill. And the idea here is that one of the interesting trends is as the, like you mentioned, we have, as antibody testing is going on, there is actually, COVID-19 testing is going on more in the community. With more testing, there is more patients who are being diagnosed in the milder setting. Patients who otherwise have mild flu-like illnesses like, or slightly severe flu-like illness, but not meeting the definition of moderate or severe COVID-19 infection. So those are the patients we're thinking about extending the study to. I think we'll make that decision. Mike, you may want to talk about that, but very soon. There are obviously other kinds of medications that are coming on board. There's a lot of different kinds of medicines that drug companies and other kinds of biotechnology companies are coming forward with. One of the challenges of doing trials in patients who are milder in course is that, like Dr. Gordon mentioned, we still don't completely understand the reason some patients actually develop severe symptoms. The people who develop milder symptoms, even sort of on the upper trajectory of the milder symptoms, seem to recover just fine on their own. So a possible benefit of using a combination like atovaquone and azithromycin might be that their duration of illness might be shorter, but their severity of illness might have not progressed to the 15% anyways, but it could still be a contribution. And just from these kinds of discussions, you can already tell that it's really difficult sometimes to tease out the actual benefit. That's where the testing that Dr. Engelthaler's group is doing for us might be really useful, because if people have, if we use milder patients to go on the trial, then what would be really quantifiable would be at what rate did their swabs become negative very quickly, did they develop the antibody response very effectively. So that's why I think we're well positioned within this trial with our collaboration here, where we are at TGen and at HonorHealth, to maybe extend this to the milder population. But yes, we're actively thinking about that.
Okay, we have a couple of questions here about how this drug combo has been used in the past in other diseases. So Dr. Sharma, could you reiterate some of the things?
Yeah, sure. So this combination, atovaquone is actually a very safe drug that is used as an anti-malaria. It is used commonly in combination with another drug called proguanil. The combination is actually used to prevent malaria. So if you're traveling to a malaria-prone country, you get, if you're going to take malaria prophylaxis, then you take atovaquone and proguanil as a tablet every day. So that's one way atovaquone itself has been used. But the particular combination, atovaquone and azithromycin, is actually used, has been approved and is used and is highly effective in treatment of a parasitic disease called babesiosis, which is a rare parasitic illness. And it's relatively safe. I mean, no drug is 100% safe, no combination is 100% safe ever, but relatively safe. So that is kind of the history behind this. Hydroxychloroquine, for instance, or chloroquine is at the end. Again, I'll let Dr. Engelthaler talk about this because I'm a little bit out of my element since I'm an oncologist. But bottom line is, hydroxychloroquine and chloroquine are not commonly used for malaria prophylaxis, or because a lot of the malaria in developing countries is actually resistant to chloroquine and hydroxychloroquine. But Dave, do you want to talk about this a little bit more about atovaquone?
But and actually, I'm not the pharmacist here, and I think Kieran may be able to speak to it better. But I think you're hitting on a point that you know, for this particular virus and this disease, you know, it's not reacting to everybody the same way, and we can't hope that we're going to find a magic bullet. But with innovation like this, we are going to find bullets, and these can be used, and we need to use them until we are able to get an effective vaccine in place in the population. So this trial here is really exciting. It's this kind of this Arizona innovation of combination of repurposing drugs and collaborations that have been in place and next gen technology that's going to hopefully give us a few more bullets to be used. But I think we all have to remember, though, that we're not all looking for the magic bullet. We can't think that we're going to find one drug that's going to treat everybody and get rid of this because this virus is reacting differently in different people. It's not used to us. We're not used to it. So we're going to see all sorts of outcomes. But hopefully we're going to see something pretty exciting with this one. Kieran, I'm sorry, I took away your...
I think it just was trying to add on to what you said and to what Dr. Sharma said. And I mentioned this in the beginning. It's used in a prophylactic setting in pneumocystis pneumonia, PCP, for in patients with who are about to undergo stem cell transplant. So that's one this combination is being used in pneumonia setting, but this is especially in those for prevention of the pneumonia in these patient population.
Okay, so you're going to be enrolling 25 patients into this trial.
Of course, the malarial setting and the PCP prophylaxis of for BMT transplants.
Okay, pardon me. So you're starting out with an initial cohort or group of 25 patients here. If all goes well with those patients and you're able to show safety and efficacy both, how do you see this expanding into the future? I think there was mention of working with other hospitals perhaps and having a group that is helping each other basically amongst the hospitals in the Valley.
Well, let me answer this a little bit beyond what Dr. Gordon already said. Dr. Gordon mentioned that obviously we would love to build an Arizona-based coalition that could use the drugs together to help patients here in Arizona. But I think Dr. Engelthaler mentioned a little bit here as well that, you know, the landscape for treatment of COVID and for prevention of COVID-19 is changing rapidly. So what is true today may not be true tomorrow, right? So I think we need to finish this trial and then see at that time what are the different options that are going to be available. And my gut feeling is that there's going to be a lot of different options. And at that point, you know, we need a reassessment to see where it might have its best chance of helping patients. It might depend on, you know, how other treatments work during the duration that others are trying. And I think also will depend on how the vaccination strategies come up. You know, as you know and read in the media, lots of different people are working around the world to develop a vaccine in sort of an accelerated fashion. So I think once we are closer to the enrollment phase of this, then we will have a better idea, you know, if it works, and if it does work, where would we be using it and how extensively could we use it?
Steve, if I may jump in and Sunil and Mike indulge me here. We developed this study not just as as a combination study for these two drugs. We developed this like a scaffolding study, like it's a basket trial, where one part of the arm that we just developed is this atovaquone azithromycin combination, but we were also looking at multiple other drug combinations and individual drugs. We even thought about a drug called ivermectin in this category, and We're also looking into multiple other drugs on this particular platform. So it's not just one study, it's a group of studies bundled up into one large study that we're envisioning. Mike, Sunil?
Yeah,
I
go ahead, Mike.
I think that what we have essentially have done is recognize that, like battling cancer, which is kind of our natural responsibilities in our roles as oncologists, Dr. Sharma and I have applied the skills that we've learned over the years in that battle towards the battle against COVID-19. And so I don't have an expectation that any one drug is going to make the definitive difference, but we need to begin the process of investigating options and doing the basic science that helps to direct us towards understanding or identifying the highest risk individuals who may ultimately benefit from either prophylactic therapy to prevent the development of the infection or earlier treatment with well-tolerated drugs. Sunil, what were you going to comment?
No, I think it's the same. I think, we're everybody in the world, it's such a difficult problem and kind of a immediate problem that we've faced unexpectedly, that all of us are putting our heads together and bringing, whatever knowledge and whatever contribution we can all make to try to help patients and each other. So like Dr. Gordon mentioned, our day jobs as oncologists have temporarily, still continue, but have also been temporarily sort of repurposed in its own way to try to come up with, different strategies to help with this particular disease. So no, I think that's well said, Mike.
Okay, the initial 25 patients, I understand that they will come from the HonorHealth system. That they would be patients that have been hospitalized for COVID-19. Is that correct?
That's correct.
Okay. And also another question, is it accurate to say that this combo shortens the duration of the sickness and lowers the severity of symptoms? Would that be your hope?
That certainly is among the goals. I'm sorry, Sunil, go ahead.
No, that's fine. Mike, sorry.
Certainly among the goals that we're shooting for, but we're only going to know that by conducting the trial and evaluating these endpoints.
Okay. Dr. Engelthaler, I'm going to throw it back to you at this point and ask you to speak a little bit about what we might be able to use the antibody tests for. Would this feed into, for example, the COVID immunity study that TGen is conducting, and also the Arizona COVID-19 Genomics Union that was announced about a month ago to sequence samples of this disease?
Certainly. Well, we're definitely trying to make sure that the number of studies that are happening that we're connected to or that are happening really around the world can really make benefit of each other. And so from this particular study, we'll be able to look at the patient samples, be able to capture, as I mentioned, the viral strain. We'll sequence those, and that'll add to the growing knowledge base about the strains that are existing in Arizona, as well as we could, and as we compare those to the global strain. So we're just going to learn more about the virus and what strains are circulating because of this study. We're also going to be looking at, because we'll capture a sample multiple times, does the virus itself generate any mutations during the course of infection? We do see some mutations in showing up, causing different strains, different lineages of the virus to move around. Can we actually see that happening during the course of infection? And we can only do that by taking serial samples, and that's part of this study as well. Taking more than one sample over time, we can monitor what's actually happening to the virus too. We're not anticipating any major changes, but at least now we can look at it.
So could the antibody tests that are being done, especially through the COVID immunity study at TGen, could that lead to a better, more accurate type of antibody test so that people could feel more confident perhaps at some point in the future of going back to work and going back out into public?
Yeah, certainly there's a number of the serology tests that are available out there. And at TGen, we've been working closely with Northern Arizona University and other partners who've been working on developing serology tests for a long time to make sure that we're using the best available now. But some of the additional work that's happening, especially through the immunology department at TGen, is to really narrow down and come up with even better targets and better serology assays that are going to be more specific and still very sensitive. Right now, there's some lacking in specificity, which means that we're going to get a number of false positives with the tests that are out there. Not for this study because it's pretty controlled, but the serology tests that are being used in general. So our hope is that we can add some knowledge to that. We're going to have some additional samples here that'll be used to validate those new tests as well. So I think just everything is improving all sorts of research and new technology development.
Okay, so I don't have any more questions here in the chat, other than let's see here.
While you're looking, Steve, I'll just, in follow-up to that point, you know, one of the challenges we have is that the currently commercially available serology tests are generally qualitative tests that tell you whether or not an antibody that would cross-react with COVID-19 is present. But as Dave mentioned, those antibodies that are being picked up could actually be to other viruses and not specifically to COVID-19. And those tests are not qualifying patients to actually donate plasma. So one of the things that we have to recognize is that as we've seen patients develop symptoms for COVID-19, I think that there are a lot of patients who took it upon themselves to simply quarantine themselves, particularly if they had known travel histories or known exposure histories to other family members. And many of those patients did not go and get nasopharyngeal swabs and therefore don't have a definitive diagnosis of COVID-19. And better antibody tests will need to come forward to be able to truly give us quantitative assessments of antibodies, because one of the challenges that is a bit concerning is whether the development of antibodies will definitively mean immunity in the future for individuals who have been exposed. And this is why we still have to be vigilant about exposure to other people and being careful about where we go and what we do. We do not yet have what we would consider to be a green light to going back to normal.
Okay, but anyone else? So in summary about that, this clinical trial that has now begun could lead not only to better treatments for patients, but perhaps also better diagnostics and other treatments such as the convalescent plasma.
I think that's all accurate.
Okay, and we have about 4 minutes left on our hour here. I was wondering if all the presenters might just take a moment to sum up, things and also mention anything else that hasn't been already talked about.
I guess I can start. I think I want to basically thank a couple of people, because in these kinds of studies, it's really difficult sometimes to get things done in time for helping patients. So I want to thank the HonorHealth Research Institute administration, as well as the foundation that has helped to fund this study, as well as TGen and its leadership and TGen's foundation, because everybody sort of stepped up personally as well as from an organizational perspective to help get this study online. Really, like I said before, we're all hands on deck trying to help patients and trying to come up with Dr. Engelthaler's term here, Arizona innovation, to try to make something happen for our patients. So thank you for Steve for this opportunity.
Absolutely. Dr. Avancha?
Yeah, Steve,
or Dr. Gordon, either one.
I was just going to echo Dr. Sharma's points. I think that this represents a community-based support for initiating these types of trials and bringing novelty and innovation to our patients here in Arizona. And while we have the hope that it's going to have an impact nationwide or globally as we begin to do our research, we understand the importance of the commitment to our community.
Steve, thanks for, to add to, and again, I echo the sentiments of what Dr. Sharma and Dr. Gordon has alluded to. I would like to take the opportunity to thank all the colleagues at HonorHealth and HonorHealth Research Institute. I mean, when this pandemic was brought upon us, 4 weeks ago, we didn't have this COVID research program. And we had to pivot as a whole research institute into getting this thing and establish the infrastructure to all the way from regulatory framework to consenting processes. I mean, how do you consent a patient when this patient is in isolation kind of a methodology? So looking at smallest of the details to the largest in the grand scheme of things, how a particular project like this has, or even projects, not just one, but multitude of other projects. We went into full gear mode. We vetted close to 30 plus clinical trial opportunities at the Research Institute. And again, our partners at TGen and coming together from a translational standpoint of view was incredible. And I want to say that the collaboration throughout this process is extremely efficient and it's what's needed at this hour. We're all working together. We're all in this together. And the support that's being done all the way from the research nurses to research pharmacists to technicians, laboratory technicians, to our physicians, to nurses, and of course the patients and their patient family members who were supportive of participating on this on these research trials. So it's overall an incredible journey and a story to share. This is not just, it's not yet completely written. We're in the midst of this as we're writing this story.
Okay, any final thoughts from anyone else?
Yeah, I think we don't get here unless we have the support of our first responders, right? It's our physicians and our nurses that are on the front lines. And while we're innovating and developing strategies, we have a great group of doctors and nurses that are actively engaged in taking care of these patients, both in the inpatient and the outpatient setting. We owe them a debt of gratitude for allowing us to bring to bear these investigational trials and collaborating with us. They're by far our greatest heroes.
Very good. I want to thank all the reporters who came on board with us today. And I just wanted to reiterate that we will have a recording of this session available on both the TGen and the HonorHealth websites. I'm not sure where it'll be on the HonorHealth website, but at TGen, you'll be able to find it by going to tgen.org/news and clicking on the press release that you all received. There'll be a copy of that on that webpage. And up high in that press release will be a link to our YouTube site where this recording will reside. So thank you all again. Really appreciate everyone's participation today and good luck.
Thank you, Steve.
Thank you, everyone.